So now that you have an understanding about the structure of lipids, and in particular triacylglycerols, we're going to start to talk about how our body utilizes triacylglycerols for fuels. So we're going to think first about the digestion of triacylglycerols by our digestive system. So the digestive system begins to break down triacylglycerols in the small intestine, where bile salts, which are made by the liver, stored in the gallbladder, and then secreted into the intestine, surround the triacylglycerols to form little bubbles called micelles. So these micelles are basically bile salts that surround the fat globules from the food that we eat. Now, there are other enzymes that are secreted by the pancreas called lipases. And lipases are going to cleave the ester bonds that were formed when the fatty acids were joined to the glycerol, the 3-carbon glycerol backbone. Now, what that's going to yield is, for every triacylglycerol, that's going to yield 2 fatty acids and 1 molecule that is referred to as a monoacylglycerol. So that's going to be the glycerol backbone with just 1 fatty acid remaining. And then those monoacylglycerols and fatty acids can be absorbed by the epithelial cells of the small intestine. So we'll talk a little bit about this through the diagram that you see here. So the small intestine is where we begin to break down the lipids that we eat. So the triacylglycerols are, again, they're going to be a glycerol backbone and then an ester linkage where fatty acids have been attached. Now, the enzymes that are present in the small intestine are going to utilize water to cleave the bond between the hydroxyl group of the glycerol backbone and the carboxyl group of the fatty acid. And again, that's going to leave us with 2 free fatty acids and 1 monoacylglycerol. And the fatty acid that remains bound to the glycerol molecule is going to be positioned at carbon 2. Now, these pieces that were generated can then be absorbed by the small intestine, by the cells of the small intestine wall. The triacylglycerol in its original structure is too large, and there aren't proteins available to transport that large molecule across the intestinal wall. So that's why enzymes have to break that down into smaller pieces first. Now, once in the epithelial cells, those pieces will be reassembled and the triacylglycerols will be regenerated. The triacylglycerols will then complex with proteins and other lipids to form lipoproteins. Remember, lipoproteins were discussed in the end of chapter 17, and that those are basically small packages where triacylglycerols or cholesterol and cholesterol esters were packaged to be delivered to the rest of the body. So these lipoproteins, which are called, the ones that are leaving the small intestine are called chylomicrons. Those lipoproteins will be leaving through the lymphatic system and then enter into the bloodstream. And then once they're in the bloodstream, they can be delivered to cells like the heart, skeletal muscle, or adipose cells or fat cells. Now, those cells can utilize those fats for those triacylglycerols for energy, where they'll break that down into fatty acids and glycerol, and we'll talk about that. Or if they are adipose cells, they can actually store those triacylglycerols for later. So fat cells are the cells that will store triacylglycerols. So they'll actually take those triacylglycerols and keep them in this storage form. And we'll see in the next lecture how efficient these triacylglycerols are at storing energy compared to the amount of energy that's stored in an equivalent amount of glycogen or glucose. So fat mobilization is going to be in response to low blood glucose and a depletion of glycogen stores. So when fats are mobilized for energy, the adipose cell is going to break these triacylglycerols down into smaller units that can go back into the blood and be utilized by other cells. So they're going to start out, again, by taking enzymes that are called lipases to start removing fatty acids one at a time. So first, there's going to be a triacylglycerol. So triacylglycerol is a molecule, this is this molecule here, 3-carbon glycerol with 3 fatty acids. It removes 1 fatty acid to leave us with a diacylglycerol. There's then going to be another enzyme called a diacylglycerol lipase that's going to take off another fatty acid. And then finally, a third lipase will take off the third fatty acid. Now that's going to leave us, for every triacylglycerol we start with, that's going to leave us with 3 fatty acids and 1 glycerol molecule. Now while the enzymes are doing that, each enzyme will utilize a water to cleave the ester linkage that is connecting the fatty acid to the glycerol backbone. So that's going to, again, it's going to form free fatty acids and glycerol. Those free fatty acids are then going to be, they're going to go into the blood. They'll complex with blood proteins to be carried to other parts of the body. The glycerol will most likely go to the liver, where it can be converted into glucose. So the glycerol from fat digestion, in a scenario where the blood glucose levels are low, the liver has the very important job of generating glucose. And the way it does that is through the process called gluconeogenesis. Now what that means is that the glycerol molecule that we achieved from the digestion of fat has to first be converted to glycerol 3-phosphate by adding a phosphate from an ATP molecule. And then we are going to oxidize that molecule into dihydroxyacetone phosphate. Now this is a molecule we've seen before, and we saw that as an intermediate in glycolysis. It can also be used by the liver to go in the reverse direction to produce glucose in the process of gluconeogenesis. So this is what's happening with our glycerol molecule. The glycerol molecule is a trihydroxy molecule, so 3 OHs. At a cost of ATP, a phosphate is going to be added down here at this carbon, or at this hydroxyl group. So here's our phosphate. We now have the molecule glycerol 3-phosphate. Now what's going to happen now is the oxidation of this molecule. So remember, oxidation, in the process of oxidation, we can take a hydroxyl group and convert that to a carbonyl group. So we're taking the secondary alcohol group and converting it to a ketone. And anytime we've got oxidation, we have to have the reduction of something else. So I've told you guys before that when we have bonds between carbons and oxygens, and we're converting from a single to a double bond, that's nearly always going to be accompanied by the generation of a molecule NADH. Now remember, NADH can be utilized to make ATP. Now this dihydroxyacetone phosphate can have 2 fates. It can either be utilized to provide more ATP through the completion of glycolysis, or it can be utilized in the process of gluconeogenesis to create more glucose that the liver can add into the blood for access, for the red blood cells and the brain to have access to that glucose for their own energy needs. We'll talk a little bit more about that when we talk about the balance of carbohydrate and lipid metabolism in class later this week. So that's going to summarize this initial breakdown of triacylglycerols, and in the next section we're going to talk in a lot more detail about what happens to each of those fatty acids as a means of providing energy to the cells of your body.